So, good afternoon again. As Pablo said, my name is Eliseba García, and I'm doing my thesis here in the University of La Laguna, and Jose Carlos Hernández and Sabrina Clemente are my PhD advisors. For the last year, I have been working on this research. It is set in a broad research project called Acid Rock, which tries to assess the impact of climate change on the Canary Islands benthic ecosystems. Human activities have become an important component in the climate system in the last century. Global warming and increased atmospheric carbon dioxide are the most obvious effects of climate change, causing the oceans warmer and acidify. The increase of seawater temperature varies the distribution and adaptability of the species, and the survival can be compromised within a specific temperature range. The redistribution of the species can produce serious problems in specific regions, causing an imbalance on local ecosystems. The temperature in the seawater in the Canary Islands has increased 1 degree from 1985 to nowadays, and it is thought that it's a tendency for the future. With regard to ocean acidification, recent global models predict that pH at the ocean surface will fall by an estimated 0.2 to 0.4 units by the year 2100, largely due to human-driven emissions of carbon dioxide. These changes will have significant effects on marine organisms, especially those with calcareous skeletons, such as echinoderms. Experts of KIMA predict for the Canary Islands an increase in ocean acidification as a result of a decrease in pH in 0.002 units by year. This study is based on the sea urchin Paracentrotus lividus. It is a common species that is widely distributed throughout the Mediterranean Sea and North Atlantic from Ireland to the north of Africa. And it is in the Canary Islands where this species has its southernmost limit of distribution. This species is not only ecologically, but also economically important. In Spain, it provides the largest economic return for sea urchin fisheries. Furthermore, it also acts as a keystone species, being an important grazer in the benthic communities. Their larvae are produced during well-defined periods and remain in the superficial layer until metamorphosis. Therefore, any change affecting their survival or growth reduces the number of individuals recruited and leads to major changes in ecosystems. Little is known about the possible long-term impact of predicted changes due to climate change processes on larvae of invertebrate, of marine invertebrate. However, in the last 10 years, there is some research that is going in this direction. Early development stages of marine invertebrates, as fertilization, embryogenesis, and larvae development, are generally the most sensitive life phases. Recruitment success depends on the embryo larvae survival, and consequently, any decrease in embryo larval survival or a delay in development can reduce population long-term viability. For this study, mature specimens of the sea urchin Paracentrotus lividus were collected by a scuba diver from subtidal rocky shores between 5 and 10 meters depth from the northeast coast of Tenerife in Boca Cangrejo Bay. Individuals were collected in March of this year during the spawning period of Paracentrotus lividus. We did two different experiments, one to see the effects of global warming and ocean acidification on fertilization, and another to see the effects of ocean acidification on the larvae development. In fertilization experiments, we had two different assays, one with pH treatments and another with temperature treatments. We used experimental ranges of pH, including current values for the parameter for the region of study. It was the control treatment with 8 units of pH and simulated predictable values of the parameter for future conditions due to climate change processes. So we had experiment with 7.7 units of pH and another with 7.4 units of pH. It is the prediction for the year 2100 and is the prediction for the year 2300. With respect to temperature treatment, we had a control treatment with 19 degrees. That is the normal temperature in the seawater in the Canary Islands in March, where the larvae is typically in the water. We had another treatment with 20.5 degrees. That is the higher than normal value that has been checked for the region of study in the last century. And finally, another treatment with 22.5 degrees. That is the prediction for the year 2100. After fecundity, we assessed fertilization rate, cleavage rate, and 3-day larvae development for pods that were incubated 50 minutes, 100 minutes, 200 minutes, and 3 days respectively. After incubation time, the development was stopped and fertilization rate was calculated by means of counting the number of eggs with fertilization membrane. To estimate cleavage rate by the number of cells in the different stages of cell division. And finally, to assess the early larvae development, we measured total length, pulsatile arm length, and body length under a microscope. The other experiment was larval culture with different conditions of pH. The larvae were distributed in 3 seawater tables in 15 2-liter plastic beakers in each table. The 3 tables had the same temperature, 19 degrees, and the larvae were fed with rhodomonalins. We had one table with 8 units of pH, another table with 7.7 units, and another table with 7.4 units. All pH conditions were controlled by a pH control computer. Every beaker had isolated conditions and a soft constant aeration with a paddle system that was moved by a micromotor. We monitored the larvae daily and for a period of a month to assess survival, development, and growth with the different treatments of pH. So we go with the results. There was a significant influence of pH and temperature on fertilization rate. Here we can see that there was a trend of decreasing at low pH. The difference was not too much at 7.7 units, where it showed a light tolerance, maybe due to the internal sperm pH that is slightly acid. However, at 7.4 units, hypercapnia can have a narcotic effect on sperm. On the other side, there was a tendency to increase the fertilization rate with temperature. Warming waters creates a low viscosity environment, and then the sperm can swim faster. With regard to cleavage rate, there was a delay of embryonic development at low pH with respect to the control treatment. This delay was higher at the lowest pH, where most of the cells only had fertilization membrane. In the temperature treatments, we could see an acceleration of embryonic development at the higher temperature, and the differences were less after more time of incubation. In respect to 3-day larvae, the three measures, body length, total length, and postoral arm length, showed that there was a tendency, a delay, at low pH with respect to the control treatment. And this delay was higher at 7.4 units. However, in the temperature treatment, we could see that the early development was higher at the higher temperature and was smaller in the control treatment. Now we go with the result of larvae culture. In respect to survival, the density of the larvae fell down the first days, especially in the control treatment and in the treatment with 7.7 units of pH, but with time it was maintained in all pH treatments. With regard to arm growth, there were significant differences for the analysis were significant for pH and time. Logically, there was a tendency for the postoral arm length to increase with time in all pH treatments. There were significant differences between the three treatments of pH. The smallest length were reached at low pH, but the higher postoral arm length were at 7.7 units of pH instead of the control treatment. Even so, the data shows small differences in the mean values. The larvae development cycle was completed within 2 treatments with the higher values of pH, while in the third treatment with 7.4 units, the cycle was incomplete. The Plutus larvae with 4 arms appeared the same day in the control treatment and in the treatment with 7.7 units of pH. While in the third treatment, the same day, the larvae were less developed. After this point in time, every phase of development had a delay of 2 days in the treatment with 7.7 units with respect to the control. Here, we can see that the larvae with 6 arms appeared at day 7 of experiment in the control treatment and at day 9 in the treatment with 7.7 units. And the same delay happened in the other phases, while in the third treatment with 7.4 units, the delay was longer. The competent stage was reached in all pH treatment, but the settlement was not reached at low pH. Here we can see the development cycle in the control treatment at 7.7 units and at low pH at the end of the experiment. Finally, we go with the conclusions about fertilization. ocean acidification and global warming have antagonistic effects on fertilization, embryonic and free-day larvae development. Fertilization and early development would be tolerant to changes predicted for the year 2100, but the prediction for the 23rd century could have dramatic repercussions. We suggest that in order to develop management and conservation strategies, research should include a wide range of taxa, the combined effect of stressors, and studies in the longer term. With regard to larvae development, Paracentrotus lividus is surprisingly robust to ocean acidification, and the larvae development on this species shows a broad pH tolerance. A significant decrease of postoral arm length and a reduced overall growth were detected at the most acidic conditions. The results suggest that for moderate pH changes, such as those predicted for the year 2100, the larvae of Paracentrotus lividus could be able to develop, whereas extreme seawater pH values, as those predicted for the year 2300, could limit larvae development, and the achievement of a competent stage would then be compromised. We have to take into account that the larvae is able to develop normally in the experimental conditions. However, a delay in development would compromise the survival or the quality of the settlement in the natural environment. Despite the clear implications of this result for predicting effects of climate change, effects due to climate change processes, further experimental research to assess larval survival and settlement under longer pH exposures and synergetic effects with other stressors would be desirable. So that's all. Thank you so much for your attention.